Thank you for joining us for our second C. elegans webinar. We're super excited to have Brian and Steph here to help us walk through the second C. elegans experiment and give us even more tips for how to be successful with these two labs in the new HBS. So Brian, turn it over to you.
All right. Thank you so much. Yeah, happy to be back. As Steph mentioned, this is going to be part 2 of our webinar series on exploring the model organism C. elegans. I am Dr. Brian L., and I'm joined here with Steph Sturm, who is in the lab and will be showing us some stuff on the microscope again here in a little bit. So the general plan today, we're going to review a couple basics on C. elegans, some stuff that we covered in part 1, but that we had some questions on and that I think is just kind of important to solidify again. And then we're going to talk about module 2 and kind of preparing yourself and then the students for what goes into module 2 of the experiment. And then at the end, I'm going to open it up to questions. And we also do have some questions that were kind of written in after the first webinar that we will discuss. So if anybody does have any questions, you can feel free to drop them into the chat or into the Q&A section here during the meeting, and I will get to those, or somebody will respond to those as we go. Okay, so recap on what we are doing here. This is the Exploring the Model Organism C. elegans experiment. This was the new experiment that was created for the 23-24 school year. And this experiment is kind of a multi-part sort of setup that has 2 different assays where students are going to end up exploring a number of different things related to our C. elegans here. So we're going to look at motility and chemotaxis, some endocrine signaling, C. elegans husbandry, and then environmental toxicity and heavy metal poisoning, which is a whole lot of different things that we kind of cover in this one full experiment. And to do that, we divided up into the 2 different modules. So we have module 1, Amazing Worms, which we discussed last week. That one, the students are going to build a maze using copper paints, and then they examine chemotaxis in the worms with or without treatment with serotonin. We also then have module 2, which is our worm husbandry and toxic relationships. And this is divided up into kind of a subpart A and B. And for this module, the students are actually going to learn how to take care of the worms themselves. So they're actually going to take charge of preparing the worms, feeding them, kind of all that general worm husbandry. And then they'll perform an assay on those worms that they are preparing. That is a heavy metal toxicity, environmental toxicity assay, where they're going to examine the survival of these worms in different conditions. Each of these modules requires multiple different class periods, so we do need to have time in there for our C. elegans to be able to grow and reproduce and make sure we have enough worms to actually do these different assays. And then there are a couple different stopping points that are in there that should allow for some flexibility as you are trying to plan everything out here. I mentioned this last time, but just a reminder that we do have video guides available. So there's a lot of great resources that we've created for this experiment. We have our traditional video guides that go over either the student section or the instructors sections, kind of break down each thing individually and show a nice guide all the way through there. And then we do have other video resources that look at just specific parts of these experiments. One of the ones we'll be talking about today is going to be on how to perform thrash counts that we'll use for measuring worm survival. And Stephanie created a really nice video that goes through how to look at these, how to monitor thrashes, and then gives you an example of how many thrashes you would expect to see in a given amount of time. Really, really great resources, and I highly recommend everybody checking those out. Okay, so a bit of review on some of the terms, because I'm going to be using these again throughout the talk today. We've got 3 main terms that we refer to repeatedly when we are talking about our C. elegans, and those are seeding, chunking, and feeding. These are really important. These are kind of important terms to make sure your students understand as well, just to make sure that there's no confusion as they are going through the instructions and kind of understanding what needs to happen. So the first of these is seeding, and seeding is basically preparing a plate that has an OP50 bacterial culture across all of it. The OP50 are the bacterial food source for our worms. And so when we seed a plate, what we're doing is we are taking that plate, we're spreading OP50 bacteria over the surface of it, growing it up to allow the bacteria to fully kind of come up and populate across this entire plate. So that seeded plate is ready for our worms to go onto it. Chunking refers to basically transferring a small chunk of agar from one plate onto another one. And this is what we're going to use to transfer worms to a new plate and basically expand out our population of C. elegans. So you can remove multiple chunks from a single source plate, and from a single plate, you can make a dozen or more new plates that have worms on them. The worms will crawl off that chunk, they repopulate the new plate, and that's how we expand out our cultures. And then the last of these is feeding. And feeding and seeding are somewhat similar, but there is a really important distinction here. Whereas with seeded plates, we are taking a fresh plate and putting new bacteria onto it. With feeding, we are talking about putting bacteria onto a plate that already contains our C. elegans. So this would be a plate that we have previously chunked onto. We have C. elegans growing on it, and we are going to add OP50 bacteria to it to just feed those worms and keep them going without chunking them onto a new plate. And here is what this looks like. So for seeding, we have our fresh plates. We are going to add 200 microliters of OP50, which is about 4 drops of it, gently spread it around on the plates, and then we let those absorb into the agar and invert them and grow them for 24 hours at room temperature. Once those plates have been made, we can store them for weeks or even up to a month in the refrigerator, as long as we just don't let the plates dry out. So I highly encourage you to seed multiple plates at once, put them in the fridge, and then you can just pull those out as you need them, and they're kind of ready to go for you. For chunking, again, this is going to be transferring a little chunk of the NGM agar that contains worms onto a new plate. So we cut out a 1 by 1 centimeter cube. You transfer that onto the new plate. We always want to try to transfer it, so we are flipping it over and putting it so that it's worm side down onto the new plate. And the worms will then crawl out of that onto the new plate. They will start laying eggs. They'll reproduce on this plate. And those will then give us this new population. We say to incubate it for at least 3 days. Really, after even an hour, you should be able to see the worms have crawled off that plate. For sure, overnight, you'll have been able to see the worms have crawled off, and they will be moving around on this new plate of theirs. Quick review of scheduling for the experiment. We say to plan for approximately 1 month to complete the entire experiment. And that would be if you are not really taking any breaks in between the different modules. So obviously we have Thanksgiving and we have a winter break coming up, which will expand out the amount of time that we would be thinking of for this experiment. But we say it's going to take about a month from when you order the worms until when you are fully done with that second of the student modules. We want you to order the worms 2 to 3 weeks before you start module 1. When you get those, you'll maintain them for about a week. You'll expand them out so that you have enough worms for the students to use. Module 1 is going to take about 3 days for the students to perform it. That's making the mazes, that's performing the maze assays, and then analyzing it. Module 2 will take about a week for them to perform it. And now a lot of this time that we say on here, this 1 month, is going to be downtime. So you will chunk the worms, and then they'll spend a week kind of repopulating these plates and getting ready to go before you use them again. And that same sort of thing applies to this module 2. When we talk about 5 to 7 days to perform it, that's realistically probably 2 to 3 different class days that the students will actually be performing the assays on. We do have these really nice worm timelines. I highly recommend checking those out before you start ordering or before you start kind of stressing about how you want to schedule it. They do kind of walk you through really nicely, kind of from step to step to step of how to go about ordering the worms, receiving them, and then the different steps for maintaining them. So as I mentioned, we want to call Edvotek about 3 weeks before you plan to start the worm. Those worms will take about a week to arrive. And then once they arrive, you've got a couple days before you really need to make sure you're feeding or chunking those onto new plates. And then every 5 days after that, you'll want to be checking them and either feeding or chunking them onto new ones. And of course, then we plan out when we do the experiments. And at that point, you'll start to prepare them for module 1 and for module 2. And we do have some really nice extended worm care guides available. So these are, again, available through all of the Project Lead the Way online portal and everything like that. 2 different guides that we have up there. One is kind of this worm scheduling guide and checklist, which is what I was just referencing. I would look through this, make sure that everything makes sense, and you can kind of understand this workflow that you're going to need to do between ordering the worms and then utilizing them for module 1 and module 2. We do also have another guide that we've created that is Stanley's School Break Scheduling Guide. And as I mentioned, with Thanksgiving and winter breaks coming up, this one is really useful for helping to plan out how to basically factor in these different breaks and when to maybe split up Module 1 and Module 2, or how to prepare the worms to survive over those different breaks. Okay, when it comes to worm care, it's important, I guess, to remember that these C. elegans can be cultured indefinitely as long as we are feeding and chunking them when needed. These are really hardy model organisms. They have about a 1-month life cycle, but they reproduce incredibly quick. So each worm that's on the plate can lay dozens and dozens and dozens of eggs. And as long as we are feeding them and keeping them happy, you'll get multiple generations kind of constantly reproducing onto these plates. And when we chunk them, then you'll end up with some adult worms, some of the earlier larval stages, potentially even some eggs that were on that chunk as you transfer them along. What this does mean, though, is that we do need to feed or chunk the worms on this weekly basis. The instructor's guide recommends feeding the stock plates every 5 days. This can be stretched out to 7 days as long as you're monitoring the worms carefully. So what I would recommend, again, depending on your schedule, would be kind of a Monday, Friday sort of schedule for the worms, where every Friday you are either feeding or chunking the worms, and every Monday after you come back in over the weekend, you check on them and make sure that everything looks good, that there's no contamination, that all the worms seem happy, the plates have not dried out, essentially that nothing has gone wrong. When we say that every 5 to 7 days you want to feed or chunk them, what I would do is alternate between the two different processes. So in a case where you need to just extend these along, where you get the worms much earlier than you expected, or you do need to stretch these over a break, I would alternate weeks of chunking and feeding. So on week 1, you chunk them onto new plates. You come in on Monday, check them, make sure everything looks good. And then that Friday, since you chunked it the previous one, you're going to now feed them. So you take your plates, you add some of that OP50 bacteria onto it, put them back into your incubator or into your box, into your drawer, wherever you're storing them, let them go over the weekend, check them. That following week now, you would chunk them again. So every 2 weeks we chunk, every other off week we are feeding. And with that sort of a setup, you should be able to extend these guys essentially indefinitely. We do have available a resource package, kind of an extended care kit that does include extra plates and extra NGM agar, bacteria, the OP50 bacto beads. So if you do need to purchase kind of an extra little bundle of worm care goodies, we do have that available as well. Yeah. When it comes to some of these bigger breaks, Thanksgiving is relatively easy because you can let them go for this week without any problems as long as you feed them or chunk them immediately before it. For winter breaks, since that is a little bit longer, the worms will be okay over the length of it, but you do need to make sure that they are pretty healthy when you are going into it. So I want you to chunk them or feed them immediately before you leave. So that last day that you are in the classroom, make sure that the classroom is not going to get too cold or too hot. I know a lot of schools will turn down the heat, and as long as it's above 16 degrees Celsius, the worms are going to be happy and fine. So we feed them or chunk them immediately before we leave. We make sure their plates are not going to dry out, so they're inverted upside down, which helps to keep some of that moisture inside, and then just leave them be. When you come back after break, so 2 weeks is kind of the longest we'd want to do this, when you come back, the worms should be good. You'll want to feed them immediately and then chunk them the following day. So we want to kind of try to boost these guys and get them going again immediately. They will probably be looking a little bit slow. You might see a couple worms that are no longer moving around, some adult worms that kind of hit that end of their life cycle, but you should have no problem getting them to go for the 2 weeks. Just know that they will take probably about a week to kind of come back again. So I would not plan any student experiments that very first week back after break unless you absolutely have to. And if you do have any questions kind of specific to that, please reach out to us and we can kind of talk to you about some specifics on that. Okay. During Module 1, so kind of preparations for Module 2, during Module 1, you are going to be using only wild-type worms, but it's important to make sure that you are setting aside stock plates of wild-type and mutant strain worms because we're going to use those later in Module 2. And we do have instructions on how to set them aside. We recommend 5 stock wild-type plates and 5 stock mutant strain plates that are set aside. Please do not forget about these after you chunk them and set them aside, though. So we need to keep feeding them or chunking them as we go through through Module 1 and then any sort of breaks or gaps that might exist before we get to Module 2. If you're going to be delaying Module 2 more than 3 weeks from when the instructions would have you set these plates up, please remember to chunk them at some point in there onto new plates. If it's only going to be about 2 weeks, maybe 2.5 weeks at the extreme end between Module 1 and Module 2, you can get by with just feeding those plates. Just make sure, again, you're checking them, you're feeding them every 7 days to make sure that the worms stay happy and well fed. Okay. At the start of Module 2, you're going to have these plates then, and these are going to be handed off to the students, and the students will now take over caring for the worms. You'll have 5 plates of the stock wild type, 5 plates of stock mutant strains, and those will then be chunked out for all of your different student groups. And it is okay for the student groups to share some of these stock plates when they are doing their worm husbandry. Again, just make sure they all look good. If you happen to notice that one of these plates dries out, the lid didn't get placed on top of it all the way, or it wasn't inverted properly. If something grows some contamination, don't worry about it. That's why we have the 5 plates of each. Realistically, all you need is one of these plates. And from the one plate, you'll be able to make enough chunks for all the student groups. But we want to have enough that you're not having this big bottleneck where everybody's trying to share off just a single plate. Let's see. Yeah, I think I covered most of this. Don't let the worms get too dry. At the same time, don't let them get too wet either. So if you feed them and you notice that it's not all absorbing in, that your plates are just more humid, they're a little bit more moist than what you were expecting, on the following time that you go to feed them, put a little bit less in. So instead of putting 200 microliters of the OP50 and spreading it around, try 100 microliters and spreading that around. The worms are okay surviving in liquid, but they don't prefer it. They'd rather have more of a solid surface that they can swim around on. And then we want to store the worm plates in the dark. We're shooting for temperatures between 16 and 25 degrees Celsius. If you go too cold, the worms will freeze. So refrigeration is bad on these guys. Similarly, we don't want to put them accidentally into like a 30 or 37 degree incubator that is too warm, and you will end up drying out the plates and the worms will all dry up and die. So we want to keep that 16 to 25 degrees Celsius temperature if at all possible. Prepare your seeded plates ahead of time. I mentioned this before, but again, big tip. You can add the OP50 to your plates, grow it up overnight so that you have these seeded plates, and then just stick them in the refrigerator until you're going to need them. And then when you need to use these, when you're going to chunk onto them, take them out of the refrigerator, let them sit on a bench for 5 or 10 minutes just to warm up a little bit so that it's not so cold when you transfer onto them, and they'll be ready to go to chunk your worms onto them. Okay, so let's talk about Module 2. We kind of did our recap. So moving on to the new stuff now for Module 2. This module has been divided up into 2 subsections. So Module 2A is worm husbandry, and Module 2B is toxic relationships. So Module 2A, we're going to ask the students now to take over this worm propagation and worm husbandry. So they're going to seed plates. They're going to chunk onto those plates, one of each for wild type and mutant strains. And the idea here is that we want to give the students this understanding of worm husbandry and kind of ask them now to take care of our model organism, which means that the plates that they are making in Module 2A are what they're then going to use in Module 2B. In 2B, they're going to do this toxicity assay, our toxic relationships assay, where they will be treating these 2 different strains, our wild type and our ADR2 mutant strains, with various heavy metals or pesticides to see if we can get an effect on the worms, and that we can then monitor it doing a thrash count assay. This is a really cool assay, and Steph is going to actually demonstrate it here in the lab here in just a second, so you can get an idea of what we're actually looking at live under a microscope. Teacher preparations for this are going to feel relatively light compared to maybe what you had to prepare at the very start of the experiment. We need to have a couple of NGM plates for the students. These are plain NGM plates, so don't seed these for the students, no worms on these. These are just our basic stock NGM plates. They need to have access to those source plates as well, so the wild type and the mutant strains. They'll get their own tube of OP50 bacteria that they will then use for Module 2A in order to do their husbandry. So that's what they're going to use to seed their plates and then chunk onto them. For Module 2B, you're going to need to aliquot out a couple different reagents. So there's environmental toxins, there's a buffer that's called S buffer that the students are going to use, and then just some plastics. And all of this can be prepared at least a week ahead of time. So if you want to prepare it, aliquot it out ahead of time, and just stick it back into the refrigerator to wait until you actually need it, that is completely fine with it. The last thing is just make sure that you are preparing your microscopes ahead of time as well, just to make sure everything's set up there. The same microscopes that you use in Module 1 will work perfectly for Module 2. Okay, so for the worm husbandry, this is nothing too different than what you were already doing. So the students are going to set up their 2 different plates, one for wild type, one for mutants. They will seed them with OP50, let that grow up, and then toss that into the refrigerator for as long as you need until you're ready for them to come back. 24 hours is all that is really needed. So you could theoretically have them set it up one day and then the following day come back and start chunking onto those plates if that lines up for your schedule. Otherwise, you stick them in the refrigerator and you pull them out as needed. For chunking, the students are going to chunk onto the 2 different plates. So they'll do a chunk of wild type C. elegans onto the wild type plate, a chunk of the mutant strain onto the other plate. And they'll need to grow those for at least 3 days. Realistically, anywhere between 3 to 7 days works really well for it. We want enough time that the worms can start to reproduce. We don't want to go so long that then the students are now having to feed these plates if we can avoid it. Okay, so Module 2B, we are going to be doing a new assay then. So this is our toxic relationships toxicity assay. What we're going to do is we're going to have the students collect wild type and mutant worms from those chunked plates that they made. They're going to use those to perform their toxicity assay. So each group will be assigned 1 toxin, which they can then compare across groups and kind of pool data across the entire classroom. They will use their different worms and this toxin to perform a thrash count assay. Within this module, what we're doing is we're trying to compare the effects of potential toxins, so different heavy metals or pesticides, on the 2 different worm strains to see if there's a difference in worms that have a mutation in ADR2. So those are our MS or mutant strain worms. It's a relatively cool little mutant. What this is a gene that is involved in RNA editing. So it's a adenosine deaminase acting on RNA, ADAR protein. And what this is doing is it's involved in RNA splicing. So normally this protein, this ADR2 protein, helps to convert adenosines into inosines, and this is involved then in RNA processing and RNA splicing. It allows a single RNA strand, mRNA strand, to be spliced into multiple different final RNAs that are then turned into different proteins. Essentially, it's increasing the amount of proteins that we can get from a single gene. These mutant worms no longer have any sort of functional ADR2. And what this means is that the protein diversity in these worms is significantly decreased compared to the wild type. Obviously, the worms are growing, so it is not a lethal mutation. And you'll notice that if you look at them, they don't seem to really reproduce any slower. They don't really move any slower. But if you were to do different sorts of chemotaxic assays with these, you'll notice that they respond differently to chemotactic signals than our other worms. So especially with volatile compounds, they seem to not necessarily be as attracted to them. And this is likely related to signaling within some of these neurons that gets thrown off in the worms when they don't have this ability to splice RNA in the same way that we would expect. For the toxicity assay, one of the cool side effects of this mutation is that the worms are significantly more susceptible to heavy metals than the wild type worms are. So the wild type worms will still be affected by the heavy metals, but in the ADR2 mutants, you'll see this much more amplified response from our heavy metals. Okay. Symptoms of heavy metal neurotoxicity in our C. elegans and humans are going to vary quite a bit, as you would expect from how different humans and a little C. elegans flatworm are. But what we do see in them is that there are a number of good effects that we can use to kind of monitor if there is some sort of toxicity that is occurring in these cultures. If we were to do really low doses of them, we'll see slower growth, shorter lifespans, delayed development in the worms, a significant decrease in reproduction. And then eventually, as we increase the amount of the toxins that are in the culture system, you'll start to see mutations occurring, cell apoptosis, and then eventually death in the C. elegans. Now, the amount of heavy metals that we are providing in the kit is actually incredibly low. So there's no safety risks for the students, but since we're putting them directly onto the C. elegans, it kind of magnifies this effect. We're essentially bathing them in these heavy metals, and they're able to absorb really rapidly into the worms the way that we're doing this culture. Okay, so actual hands-on part for the students. They are going to be trying to wash the worms off of these plates to collect them. And the way that we're going to do this is we're going to use S-buffer and just a bulb transfer pipette. And I do recommend using the bulb transfer pipettes that we include. I know you all have the adjustable volume micropipettes. The transfer pipettes work really nice for this, though. So we're going to add 1.5 milliliters of S-buffer onto the plate, tip it to the side a little bit, and then just gently rinse the worms off by kind of sucking up and squirting this S-buffer gently along the surface of the plate. So we don't want to spray it hard. We're not trying to dig any of this NGM agar off of it. We just want to gently wash the worms off. Rinse it a couple times, let them collect down at the bottom, and then transfer these into one of the Eppendorf tubes that are provided. So they'll do this for both plates, one for the wild type and one for the mutant strain. And we're just going to set these aside and let the worms settle down into the bottom of the tube. So we don't want to centrifuge the worms. They're not as sturdy as like a cheek cell or a bacterial cell or something. We're just going to let gravity do its work for us and let these settle down to the bottom of the tubes. While those are settling, the students will prepare the rest of the materials they need. There's some counting chambers, some plastics that they're going to use. They will figure out which toxin is assigned to them or which toxin they choose. And they're going to prepare tubes that have some of this toxin in it. The toxins that we are using, again, as I mentioned, are relatively safe for us, especially at the doses that we are using. So these heavy metals are things, zinc, cadmium, and then we are going to be using a pesticide that we provide as well. So each group gets assigned one of these different things, prepare those all out, and then once the worms have settled to the bottom, we can prepare them for the toxin assay. Very carefully remove as much of that supernatant as we can. We say to remove about a milliliter. The more you can remove, the better, but we don't want to disturb the worms that are at the bottom of it. So I'd rather leave a little bit extra supernatant if it means that we don't suck up the worms. We want to have those down there for us to use then for this toxicity assay. The next part we want to do one at a time. So we have our worms collected, our wild type and our mutant strain. They're fine hanging out in this S buffer for half hour, even longer if we need to. So don't worry about moving on to this step immediately. It's better to do things accurately than to go really quick through these next steps. So what we're going to do is we're going to have the students take just their wild type worms, gently tap the tube to resuspend them. We want to get them now back into that remaining supernatant. And then the students will transfer 20 microliters of that worm suspension into the environmental toxin, quickly mix that, and then add it to their counting chamber. We want to focus on our counting chamber. You'll see these little grids, and we'll have Steph show that in just a second here. I think she's ready to go. And then the students are going to perform their thrash count. And what they're going to do is they are going to find a worm that is moving around actively, and they will spend 30 seconds counting how many times this worm thrashes up and down. So for a thrash, we're looking at kind of an up and down motion. And each time it does that full motion, we consider that to be one thrash. So we'll count it for 30 seconds, and then we wait 2 minutes. Repeat that 30-second count, wait a final 2 minutes, and count one more time. So we're collecting time points at essentially 0 seconds, 2 minutes, and 4 minutes, and doing these thrash counts kind of over and over again to monitor the effects of this heavy metal over this kind of relatively short time period for our worms. Once you have done all of that, and you've collected all 3 different time points for your wild type worms, then we go back and we repeat steps 11 through 18 here with the mutant strain. So resuspend the mutant strain, add those to the environmental toxin, perform the thrash counts. Let's see, Steph, are we good to go in the lab here?
Yeah, I can share my screen really quick.
Okay, so I'm going to stop sharing here if you want to share yours then.
Yeah, So this is in one of the counting chambers that you'll receive in your non-perishable box for 456. And I've just pipetted 10 microliters of the worm suspended in the S buffer, which Brian just went over how you would kind of collect those worms off of the plates. And then you can kind of just see them thrashing around in that S buffer liquid, which is just thrashing is that curvature movement that you see them kind of wiggling around.
Yeah, and so any of these would be a good candidate. We are looking for basically just picking only one. If you do have a microscope that has a screen where multiple students can gather around it and observe the screen at the same time, you could have them instead each count one worm on their own. Otherwise, you can have students take turns counting at the different time points. However, that kind of works out best for you. There are also multiple wells in the counting chamber, so you can do replicates of these if you want as well. There's kind of a lot of flexibility for how you want to do it, but we want to essentially pick just one worm and do the thrash count on that one worm. Let's see, we're looking for adult worms. Almost all of these worms that you see, the larger ones that are thrashing around on this are the adult worms. If you look close, so kind of right off to the right now of dead center, there is a couple smaller worms. We're not too interested in those. The bigger they are, the easier it's going to be to count, and you're going to get more accurate thrash counts off of these adult worms. Yeah, so these look pretty good. So we'll count for 30 seconds. We wait 2 minutes, and then we do a replica again, wait 2 minutes, do it one last time. One thing to take note of, depending on the microscopes you're using, some microscopes have light directly underneath the platform that the slide, this counting chamber slide is going to be sitting on. Sometimes those can heat up pretty rapidly. So I would check that out on your microscopes and just see if that's the case. These ones that have more incandescent bulbs in them, mercury bulbs or that sort of a thing, halogen bulbs, those can give off a lot of heat, and that is not great for our worms. So as Steph mentioned, she put 10 microliters in there. It's a very small volume. So you can imagine that if it's sitting on a hot surface, it's going to heat up pretty rapidly. What I would do in that case is you put your worms on it, monitor them for the 30 seconds, and then in that 2 minutes in between, just turn the light off. And then when it's time to do the next thrash count, turn the light back on, do your 30 seconds, turn the light off again. And that will kind of help to prevent that heat building up too much on them and prematurely killing them. And yeah, so if we watch, you can see that some of these worms are definitely starting to slow down here as well on the slide as I've been kind of chatting, as they are starting to kind of slow down, and eventually they will die. And what we see when they die is the worms will turn into sticks. So they're no longer these nice S-shaped or C-shaped worms, but instead they will end up being straight little worm sticks. And that is when we know that they have died. Perfect. All right, thanks, Steph. So share my screen here again. Okay. And from this, you'll be able to pull a pretty cool amount of data from the class. So up here, we do have a control solution that is optional. So as I mentioned, we have plenty of worms and we have plenty of counting chambers. If you would like to have control worms that are just in S buffer, I definitely encourage you to have the groups collect that data. And what we can see in the top left here is that there is really no difference that we see over that time. You might see the numbers drop off very slightly, but you shouldn't really see the worms dying off in just the S buffer. But when we look at some of these heavy metals, then we should see a much, much larger difference. There again will be a difference between the different heavy metals and then also between wild type and mutant strains. So you can see in some of them, cadmium is very, very effective at killing off the mutant worms compared to some of these other solutions that are not quite so potent. The pesticide solution is not going to have as big of an effect on the wild types as it does on the mutants again. And when we graph this out, you should be able to see kind of these nice different trends in there that the students can kind of collect. And again, I highly encourage you to have the students share the data across different groups. or compare replicas of the same toxins across different groups as well. Okay. Final thing then, what do we do with the worms now that the assays are done? As I mentioned earlier, the worms can be cultured indefinitely. So as long as you're feeding and chunking them, you can continue these cultures on more or less indefinitely. They do make wonderful model organisms for other experiments or extension activities. So if you want to use them in a capstone or independent project for interested students, you will have them available. You can continue these cultures along, have the students continue the cultures along, and they work out really well for that. If you are done with them, to dispose of the worms, we recommend adding just 10% bleach solution to the plates, let it sit for a minute or two to make sure everything is dead, and then they can be disposed of in standard lab trash. These are non-pathogenic. They are naturally found in the environment, but we don't want to introduce these different strains just into the wild. So I would recommend bleaching them or autoclaving them if you have an autoclave ahead of time. Heavy metal waste can just go into standard lab liquid waste and be disposed of in that way. Okay, and with that, if anybody has any questions that you want to drop into the Q&A or chat, we are happy to answer those. I do have some questions that were written in between the last webinar and when we announced this one. So I did want to go through those as well, but if I don't answer, again, please feel free to drop anything in here. Let's see, when should I feed the worms that were sent to me? So when you receive those worm plates, we recommend checking them out immediately under your microscopes. Make sure that everything went well with shipping and that your worms are looking happy and healthy on the plates. At that point, you do have a couple days before you need to feed them or chunk them. But I would try to do that within the end of the week that you receive them. So if you receive them on Monday or Tuesday, try to feed or chunk them by Friday, if at all possible. If you receive them on a Friday, you're probably okay to wait until Monday and then start kind of the process at that point. Let's see. I want to get my worms early because I'm worried something will go wrong. Can I do that? The answer is yes. You can absolutely get the worms early, but I would consider first how early you actually need to receive them. It is only going to take us about 5 to 7 days to get the worms out the door to you, depending on when you order them and when that falls during the week. We obviously don't want to ship them where they would arrive on a weekend, but give us about a week to get the worms into your hands, which means that I would not order them too far in advance unless you really are excited about culturing them yourselves, because otherwise you are going to end up having to feed them, chunk them. And it does just introduce potentially more chances for contamination or for something to go wrong with them. So yes, we are happy to send them to you ahead of time, but I would probably consider waiting until you actually do need to order them. Let's see, what is the proper way to dispose of my worms? We discussed. Should I feed the non-dosed and dosed plates during module 1? And the answer there is no. So we do want these to be starved plates. And that is spelled out in the instructor's guide, that you are going to be treating these with serotonin on a starved worm population. And this is important because we want them to be hungry so that when the students are doing module 1, this 𝐏𝐚𝐧𝐞𝐿 assay, they really will respond to these different chemoattractants, and they'll be really excited to go and find something to eat. Let's see. Should or could I feed worms with cold OP50 culture? The answer here is probably yes, but I would recommend taking that culture out of the fridge and letting it warm up for a couple minutes before you end up feeding the worms. Because your worms are on these plates and they're already at room temperature, it's probably fine to add these couple drops of colder OP50, but they're going to be a little bit happier with you if you let it warm up for a little bit first. Let's see. How do I know when to chunk the worms? This is a great question, and this is something that you'll figure out after you've chunked them a couple times. When we're looking at the plates and we're trying to decide between just feeding or chunking, what we want to do is really just get an idea of how many worms are on it. And so if you plop this on your microscope, a plate onto the microscope, and in that field you see 1 or 2 worms crawling around, they look happy, they look healthy, and as you scan around in it, you can see a couple worms in every field. Those guys are fine to feed, and I probably would not chunk them. If you look at the plate and you see a dozen or 2 dozen or more worms immediately on that field when you're zoomed in, that's probably a time where it'd be okay to end up chunking it onto a new plate and let those start dividing out. And really, it's just kind of getting a feel for how many worms are on each of these plates. We don't want to chunk them too early, because if you are only chunking a single worm across onto it, it's going to take a lot longer for that worm to reproduce and for you to get multiple generations growing up on the plate. The last question I had written in was, I don't see any movement on my plates. Are my worms dead? The answer here is almost 100% of the time, no, unless you did something that will actively kill them. So this would be freezing or refrigerating the plates or heating them up at 37, or if the plates have completely dried out. In those cases, yes, your worms are probably dead. In all other cases, though, more than likely, no, the worms are not dead. If you've neglected them and it's been a couple weeks since you last fed them, the worms will dig into the agar. So they like to normally be on the surface, but if they are really hungry and really searching for food, they will start burrowing into it. And so it's very possible that your worms have now started to burrow into it. So I would adjust the focus on your microscope and see if you can see stuff that's crawling around inside the agar. If that's the case, you're okay. Chunk those onto a new plate, give them a week or week and a half for them to come out of the agar, start feeding and start reproducing again. And they'll be okay in those sorts of cases. The other big thing is that although you can see these by eye, if you look really close, you do need to be using a microscope to view them. So if you're looking at the plates and you can't see them, make sure that you're zoomed in enough that you would actually be able to see the worms crawling around on that surface. And with that, I think this concludes our second webinar series or webinar in our series here. Steph, do we have any other questions that you had?
No, I don't think anyone chimed in.
Okay. Sounds good. All right, so I think we are done, Steph.
Yep.
But right, awesome. Thank you again for getting on and answering all these questions. And we'll make sure for folks on the call, we'll get the webinar link out to you so that you have it. We'll also attach it in the teacher guide with the other resources for these two labs, along with the link from the first webinar, so that you have all of this great information.
Perfect. All right.
Yep.
Thank you, everybody. Reach out to us if you have questions and have fun with the experiment.